I am Mrs. Sloan, and this is a video for my AP Biology students. And this in our series on genetics and Mendelian patterns of inheritance is video number 2. So down in the descriptor of this video are the notes that my students would use, which you are welcome to use as well. I'm going to make myself a little bit smaller and get going here. All right, so we have already discussed previously in video 1, we have gone over the history of Gregor Mendel. We also went over 5.3 in the unit guide, which connects genetics and heredity and evolution. I encourage you to look over that. We have already done monohybrid crosses, and we have talked about the law of dominance and the law of segregation. Where we're going to be picking up is in 11.2 Mendel's laws, and we're going to start at the law of independent assortment. Before I get going on talking about the law of independent assortment, though, I want to do one thing first, and that is a quick review of some math, because when we start looking at our data now, we need to be able to analyze that data using our chi-square test. So I need to review that quickly with you. Okay, so this is a little rewind. You should already know this, hopefully. All right, so the difference between a research hypothesis and a null hypothesis. A research hypothesis is looking for a connection between your independent and your dependent variable from what you're choosing and what your data is. So that might be if I give my plants more sunlight, then they will grow taller because they need the light to generate sugars. Okay, that is a research if then because, making a connection. A null hypothesis would say there's no connection between the amount of sunlight and how much your plant grows. It's just due to chance that your plant happens to grow. Usually in science, because it's hard to control all the variables, you stick with the null hypothesis, which you say, hey, there's no connection, but if you have overwhelming evidence to disprove that in your data, and how do you know it's overwhelming? You analyze it with like a chi-square test to tell if it's overwhelming. Then you can disprove the null and support the research. Hopefully that is a review for you. In genetics, just like other experiments, we always go with the null usually. So you're saying, hey, the data I'm going to get, I'm going to predict it based on these are my 2 parents. This is what I predict I would get in my offspring. And so that would be my any variable from that would just be due to chance. All right. So chance is going to be a part of every experiment. For instance, right, if you had 4 kids, right, what are the odds that you're going to have boys and girls, right? It's a 50-50 chance each time. So you would predict if you have 4 kids that you would expect 50% of them to be boys and 50% of them to be girls. Well, what if all of them are girls? What if all of them are boys? Is that due to chance, or is there some other variable that's coming into play? So you're analyzing the data that you have. Clearly, if you have more data, like if you had 15 kids, go for it. Then you could say you could analyze that data a little bit more because you'd have more points, right? So when we do this with genetics, we're always going to default to the null hypothesis. And because we'll be able to know and predict what we think we're going to get. If our data doesn't support that, then we need to go, well, it must be some other form of inheritance. And that would be your alternate hypothesis. So let's go with some practice here. Let's say these 2 parents had 4 kids. And I know I picked a bad picture here because probably these 2 parents did not have these 4 children, but maybe. So if you're going to have 4 kids, you would expect 2 boys and 2 girls. So that would be your expected. But what is your observed? There are 3 boys here and 1 girl. So is that just due to chance? Is that normal? And I know I'm starting off pretty simple, but I want to elevate it in just a little bit. So I'm going to start with an easy one. So what you would do is you would write down what you expected, 2 boys, 2 girls, and we observed 3-to-1, right? Because when you have 4 kids, you would predict biologically you would have 2 of each gender. So this is where we need to analyze, is anything off here? So we use a chi-square test to determine how off are we at what chance would it be that the data we would calculate for our chi-square test is off so much, the chance of it occurring is so little that there has to be some other factor. So what you do for each of your data points, for each of your categories, you take what you observed, minus what you expected. So basically the difference between what you expected, and you square that value and put it over your expected, and you do that for each category. And you just add that up, you sum it up. So let's try that with this family right here. Okay, so here's our observed and our expected. So I observed that I had 3 boys. I expected 2 boys. So it's 3 minus 2, right, which would be 1. 1 squared is 1 over what I expected, 2. So that would be a half, right? I do the same thing for the girls. I observed 1. I expected 2. Right? I'm using this formula right here. 1 minus 2 is minus 1, but when you square it, it's still a positive value. So that's 1 over 2, which is 1/2. So that would be 0.5 plus 0.5, which equals 1. That is your chi-squared value. That is this right here. Don't do anything else to it. Don't square root it. Chi-squared, it just is. So I need to look at this look at this number on a table to determine, is this a significant number? Is there something else coming to play, or could this happen in a family? So what you do is you have degrees of freedom, and you have a probability table, and your degrees of freedom are always how many choices you have minus 1, right? Because if I'm not a boy, I'm a girl. So really it's just one choice, right? So on here in a binary world, I know there's issues with gender, just in a binary world. So I have boy and girl, so I have 2 choices minus 1. That means I have 1 degree of freedom. So the only row I need to pay attention to is this row right here. And I find, where does my chi-squared value fall in? So I'm looking across this row and I'm looking for the number 1. So here's 0.02, 0.15, 0.3. The number 1 for a chi-squared value would fall, I would predict between 20 and 50% of the time is where that would fall. And you know what? That is acceptable. If I had a larger chi-square number, if I had, and this is the cutoff point here is 5%, usually it depends on what you're doing, or if you're doing drug trials for COVID, you might want to have a different value here, but this is your cutoff right here. If your chi-square value is 3.85, that's saying statistically it's probably not going to happen. Now notice if you have more degrees of freedom, right? If instead of had 2 choices, we had 3 choices, right? Then you have a larger value here before you hit the poor fit. So I'm not worried about this family. 3 boys, 1 girl. Chances are that could happen. What are the odds it would happen? Between 20 and 50% of the time. Okay, let's just level it up and look at this family. What do you think? So you would count up how many kids they have. How many do you think are boys? How many do you think are girls? Is this, is something at play here? Or are there numbers? So you could pause the video right now and just try it on your own. I'm going to show you how to do it as well. Okay, so pause if you want to try it and identify it. Okay, I've done it here. I put G where I thought they were girls and B where I thought they were boys. And I said, okay, they had 10 kids, so I'd expect 5 boys and 5 girls. By my guess, they had 3 girls and 7 boys. So is this statistically significant? Again, so I do observe minus what I expected squared over what I expected. Do the same thing here. So hopefully this is just a review, and my chi-square value turns out to be 1.6. If you remember from before, we have 2 degrees of freedom, right? So the number where I would be concerned is 3.85. And what I got when I analyzed this family is their inheritance. This is not a problem because 1.6, that would happen around 20% of the time. And so that is, I don't need to reject my null. I can accept the null of inheritance, that they're still following the chance of boy, girl, that the sperm is 50% X, 50% Y. There's nothing odd here at play. I don't need to look at alternative hypothesis. I can support my null. All right. Now, in this case, just to review also again, 5% is your critical value. So if you predict for a cross 3 to 1, right? So if you had 100, you would predict it should be 75-25, right? If it was a 3 to 1 prediction for your cross. If your data shows it's 70-30, is that okay, right? You would have to take observed minus expected over what you expected, square it. Is that data okay? Feel free to run that. I think it looks pretty good. All right. I think that is within the realms of probability there. A behavior lab you may or may not have done with roly-polies is you could have had two chambers for the roly-polies to move back and forth, a dark and a light chamber. And if you were doing a null hypothesis, you would say light does not affect roly-poly behavior. I would expect if I have 50 roly-polies, I'd expect 25 in the dark and 25 in the light. Well, it turns out I got 40 and 10. Okay, so if you do observe minus expected here, that is going to be a very low probability. So in that case, I would reject my null and support an alternative hypothesis. In this case, that roly-polies prefer the dark. And feel free to do the math on both of those for a review. All right, so let's try it with a genetics problem, okay? A certain reptile, and this is just simple dominant recessive trait, in a certain reptile, eyes can either be black or they can be yellow. Two black lizards are crossed, okay? Two black-eyed lizards are crossed, and the result is 72 black eyes and 28 yellow-eyed lizards. So what is your null hypothesis? How do you think this is inherited? What are your expected results? What did you think you would get based on your null hypothesis for black and yellow eyes? And then test it with your chi-square. And I encourage you to pause this video right now and try this on your own. I will certainly give you the answer, but try to do this on your own. Okay? All right, I'm going to start talking about the answers now. So in this case, I would assume when I read something like this that black eyes are dominant over yellow eyes because you can't hide that dominant trait. So I had two black-eyed lizards, but they were hybrids. They were each carrying, they were like big B, little b. So the little b would represent the yellow allele. And so if I were going to do this, my hypothesis would be simple dominance and that these two parent lizards were hybrids, and I would have predicted a 3 to 1 ratio in the offspring. So what were my expected results? I made this simple for you. 72 plus 28 is 100. So I would have expected a 3 to 1 ratio, 75 to 25. Okay. Now, what did I get? Observe minus expected, right? Calculate your chi-square and see, is this acceptable? Is my null hypothesis still supported? So I predict, I did the math here for you. There are 100 total lizards. Parents are heterozygotes. I predict 3 to 1. So I would expect 75 and 25. What did I get? 72. So 72 minus 75 squared over 75. That's 0.12 if you look in the bottom left-hand corner. And then I got 28 yellow, so 28 minus the 25 that I expected squared over 25 is 0.36. I add those up together, and that is 0.48. That's my chi-squared value. So how many degrees of freedom did I have? Well, I had two kinds of offspring, right? I had black-eyed and I had yellow eyes. So your degrees of freedom is always 1 minus how many choices that you have. And so I would have 1 degree of freedom. So 1 degree of freedom, and I'm looking for, was that, I'm sorry, I already forgot. Is it 0.48? Of course, it's hiding that. Yes, 0.48. So I go across with 1 degree of freedom, and I would expect those results, statistically speaking, about 50% of the time, So I do not need an alternative hypothesis that something else is coming into play because this seems to fit the mold. All right, so that's how you can use math to analyze your results of your crosses, because you could get asked that on an AP exam. This is the cross. What do you expect? What did you get? Could there be something else that is affecting that inheritance? All right. So we have looked already previously in a video on the possibilities of one trait. So now we want to analyze the interactions of looking at two traits combined together, like dark hair and dark eyes, right? I'm just not looking at our eye color. I'm just not looking at hair color. I'm combining those two, and what are the odds I would get that in my offspring? So if you look on your notes, this is where we're going to go in the notes. So law of independent assortment, this is under 11.2. In summary, I want you to think about metaphase 1, right? They meet in the middle at a metaphase. In meiosis 1, you're separating homologous pairs. In meiosis 2, you're separating sister chromatids, right? So if the dad had the little b and a big E, and then during the S stage of interphase, it replicated, so you make your sisters during the S stage, synthesis of DNA. So now he has these two E's. These came from the dad, and these came from the mom, big B, the mom that made this zygote, right? Okay, she had big B and little e. If they line up in this way, right, and they separate from each other, then you would expect little b with big E and big B with little e. But they could have lined up a different way. Instead of lining up this way, they could have lined up this way. So you could flip that, right, because of the law of independent assortment. And so if they lined up this way, then you would get little b with little e and big B with little e in your gametes, right, depending on how they separate. So when you analyze that, what I would recommend that you do is you could use FOIL. Do you remember FOIL from math? First, outside, inside, last. So I just put right here, I put the B's, right? This individual has a big B and a little b, and just usually you always write the dominant letter first, right? So they have a big B, little b. They also, for this pair of alleles, it's also a hybrid, big E, little e. So what are all the different combinations I could get in the gametes? Well, if you do FOIL, I could get a big B, big E gamete, right? I could get a big B, if I do first, right? Outside, I could get a big B, little e. I could do inside, little b, big E, and then the last would be little b, little e. This is a much larger Punnett square, isn't it, right? Instead of just 4 squares, right, you're going to have 16 squares, right, 4 by 4. It's a much bigger Punnett square. Now, I am going to show you the long version of making the Punnett square so you understand how it works, and then I'm going to show you a super shortcut, and you'll never make a ginormous Punnett square again. All right. So first in your notes, I want you to put in summary underneath think metaphase 1, each pair of factors or alleles segregate, assorts independently of the other pairs. So the B's assort independently of the E's. All possible combinations of factors can occur in the gametes. So then let's do a dihybrid cross. All right. So, not quite yet. All right. So if we're looking at the trait of widow's peak, okay, a widow's peak would be if your hair comes to a point right here and versus a straight hairline across, and short fingers versus long fingers. What are the odds that you're going to inherit them together? So a widow's peak is dominant over a straight hairline, and short fingers are dominant over long fingers. So if you have these 4 chromosomes right here, if they line up this way, you could get then widow's peak and short fingers, or you could get straight hairline with the straight hairline with the short fingers. So you have 4 different options. So what I would do in any kind of situation like this to get these 4 is I would just do FOIL, and I will show you that one more time. Okay, so let's look at this cross right here. Here's one parent, and this is the one that's similar to the one that's in your notes. Okay, for peas, having round peas is dominant over having wrinkled peas. So that's the R's. Yellow peas are dominant over green peas. Now, I know if you go to the grocery store, you're not going to find any yellow peas, okay? It's just that green peas are what we prefer to eat, I guess, that color. And so just because a trait is dominant doesn't mean it's most prevalent. For instance, I'm betting you have 5 fingers. That's actually a recessive trait. This to have 6 fingers is a dominant trait. Okay, but I bet most people have 5 fingers. So even though it's recessive, it can be the most prevalent. All right, so if we look at this individual, it's not like we need to do a lot of FOIL right here, right? They're either going to give a big R, they're going to give a big R and a big Y every single time. It doesn't matter if you FOIL it. You'll always get a big R and a big Y. And this particular wrinkled and green plant will always give a little R and a little Y. So a mating between these two will form a dihybrid. So all the offspring would be big R, big Y, little y. Both the dominant alleles coming from this parent and both the recessive alleles coming from this parent. So now these would be the parent generation. This would be called the F1s. So now this is a dihybrid cross, crossing two of these together. So first you have to identify the gametes, right? And this is where you would do your FOIL. So just pause for a second and see if you can figure out the different gametes for this. Okay, and then here we go. So if I did FOIL, I could get big R with big Y, big R outside, big R, little y, inside, little r, big Y, and last little r, little y. And I could get the same thing for this one. So now I need to make my Punnett square. Okay, so here's my Punnett square, and I'm crossing the F1s. So I would need to put a big R, big Y, big R, little y, little r, big Y, little r, little y across here and also here. Now, why aren't you going to get an option of two R's together and two Y's together? There's a law, right? The law of segregation. You will separate R's from one another. So in the gametes, there is only one version of the R and there is only one version of the Y, and we have to give all our possible combinations of gametes to predict what we're going to have here. Remember, these would be your resulting offspring or your zygotes. Okay, so I would line up all of my gametes right here. Okay, and then what I would do is I would cross them, and I've done that for you already. Okay, and so here you can see if I put all the gametes here and all the gametes here, I've got big R, big Y, big Y. You can see all of those. You can check it, make sure I did it right on here, okay? So these are all the different genotypes. But now we could figure out what are all the different phenotypes that I could get from that, right? So for instance, big R, that would be round, and big Y would be yellow, right? So the phenotype of this organism would be round and yellow. This one would also be round and yellow. Let me give you a little tip. Because one of the gametes has the two dominant alleles, all of these will be round and yellow. And because this gamete has the two, all of these will be round and yellow. And so we could go ahead and we could start to fill each of these in with what it would look like in the phenotype, right? So round and yellow, all of these are round and yellow. Okay, these two would also be round and yellow. You can go back and here, you can look right here. See, round and yellow, round and yellow. All right, so now once I've done this, round and yellow, that is the dominant trait. 9 of these 16 offspring are going to be round and yellow, right? So then now let's look at the next one, round and green. So this one right here has at least one big R, so that makes it round. The two little Y's would make it green. That's the recessive trait. So this would be round and green. Can you see some other ones that would be round and green? I'll let you look. Okay, these 3 would be round and green. Then the next one is round and yellow. So let's try to see how many rounds and yellows I have. And so I look and I have 3 round and wrinkled. Sorry, I didn't mean to say round and yellow. Round and wrinkled. Sorry about that. And then, so these are my 3 round and wrinkles. And then this is the only one that is going to be wrinkled and it's going to be green. Okay, so there's my wrinkled green. So what is my phenotype ratio here? I have dominant, recessive, dominant, and recessive, right? And so if you count that up, you will see that the ratio is 9:3 to 3:1. When you do a dihybrid cross and it comes out 9:3 to 3:1, that's like evidence, yes, that is a dihybrid cross. Now, I'm going to put a pin in something that we're going to talk about later in the next video, and that is linked genes. Linked genes, if the R and Y were on the same chromosome, they wouldn't assort independently, right? Because they're on the same chromosome. So if big R and big Y is on the same chromosome, they're going to travel together. And so you wouldn't see this type of prediction here. If you got something from 9:3 to 3:1 in a dihybrid cross, I bet every time it's going to be linked genes. So just put that in your head already, okay? This would be typical of a dihybrid cross. Now, what if you had to do a trihybrid cross? Now it's getting even bigger. Like, wouldn't that be 64, I believe, is how many squares you would have to make. You do not want to do that. So what you want to do is learn the shortcut so you don't have to make the big Punnets. And the shortcut is this. Do individual Punnets for each of your traits, and then use the product rule that we learned about last video and just multiply your results together, and you can figure out anything you need to figure out through the individual Punnets. And pretty soon, you won't even need to draw the individual Punnets. You will see it in your mind. You will see it. You will smell it. You'll like, that's what that Punnett square would turn out. You can just do the math for it. Okay, so let me give you an example of that. So the same cross as we did before. Big R, little r, big Y, right? A dihybrid cross. But this time I just make the Punnett for the R's. So I cross just the R's together, big R, little r, big R, little r. Okay, and I do a Punnett just for the Y's. And then I want to know, what are the odds that I would get round and yellow? When we did the big Punnett square, right, we said 9 out of 16 we're going to be round and yellow. Does that show itself here? Well, what are the odds of roundness out of this Punnett, right? 1, 2, all 3 of these would be round, right? Only 1 would be wrinkled. So 3 out of 4 would be round. And over here, 3 out of 4 would be yellow. So all that you need to do is take the probability of the roundness and multiply it by what are the odds of yellowness, right? In our product rule, 3 times 3 is 9, 4 times 4 is 16. I would predict 9 out of 16 would then be round and yellow, right? And then let's go again. Let's do another one. What are the odds of being wrinkled and green? Well, what are the odds of being wrinkled? Well, 1 out of 4. What are the odds of being green? 1 out of 4, right? 1/4 times 1/4. 1 times 1 is 1. 4 times 4 is 16. So you don't have to do those. And I could throw on another trait. I could have thrown in S's and have you do that, and you could have figured that out. Any genotype that I gave you, where it be big R, little y, you could figure that out, right? Big R is 1/4. Little y is 1/4. So 1/4, right? Same idea, 1/16, that you would get that particular genotype, all right? That is your product law. All right, now, if you wanted to combine what I've taught you about chi-square and product law, you could use Indian corn. Now, in this case, Indian corn, being purple and smooth is a dominant trait, okay? And then purple wrinkled would be a dominant with a recessive. That would be this, right? And then yellow and smooth, that would be another recessive dominant. And then yellow and wrinkled, that would be the recessive trait. Okay, so again, this is the same scenario where you would expect 9 to 3 to 3 to 1 in each of these. So you could analyze this Indian corn and count up each one and tell what you would expect, and then take the difference on these 4 different categories. Now, that takes a while. I did do it for you. I'm just going to show you that I did that. And so I identified what each one was. I counted up how many I had total. Now, what I would do is if I counted up 70, right, I would say 9/16 times 70, I'd expect 39 of the dominant, right? And then 3/16 of 70, I'd expect around 13 purple and wrinkled and 13 yellow and smooth, and 1/16 of 70 is 4. So the purple over here is what I expected, and then this is what I observed when I counted it. So then I would just do a chi-square test on that, right? So here's my expected numbers. Here's what I observed when I counted them. Observed minus expected squared over what I expected. I do the math for all of that. I added it up and I got 5.76 for my chi-square value. Now I need to know, is that chi-square at 5.76, is that a concern? Is this normal Indian corn, or is this some sort of mutant Indian corn? Is there some other factor? So then what you would do is you would just look on your table, right? So I had how many different categories? I have 4 different categories, right? So take away 1, that means I have 3 degrees of freedom. So I'm going to need to look at 3 degrees of freedom and look up the number 5.76. So I'm going down to degrees of freedom. I'm in this row right here, and I'm looking up 5.76. I'm coming on. Oh, am I going to make it? Yes, I am. okay? Because the cutoff for 3 degrees of freedom is 7.82, because if I got a number, if I had deviated that much, that I had a chi-square value of 7.82, if I had deviated off my prediction that much, then that would be a poor fit. There was probably something else coming into play, right? But my number of 5.76, it falls somewhere between 10 and 20% of the time. So I can still support the dihybrid inheritance. I can still support my null hypothesis, all right? Okay, there are some practice problems my students will be doing in class. I'm going to leave them here for you. If you're interested in doing these practice problems, go for it. I'm happy to put the answers in the descriptor of the video. And next, what I want you to do is, we are on 11.3, Mendelian patterns of inheritance and human diseases, and we are going to talk about pedigrees. So not just dog food. So when you look at pedigrees, you will see that the female is a circle and males are square. This is referred to as a marriage line. I guess we could say partner line, but that is how they first named it, is this is the marriage line. Then whatever comes down from the marriage line, this is the children. So they had a girl. Typically in a pedigree, what they'll do is they will color in the affected, whoever has the disease or whatever it is you're tracing. So in this case, both parents were fine. They didn't have the disease, but the child did. So how do you think that was inherited? Would that be inherited as a dominant trait, as a recessive trait? And later we'll bring in the option of a sex-linked trait. We're just not quite there yet. Now, I'm going to tell you, when I look at this pedigree right here and I think, okay, neither parent had it, but they had a child that did, they were probably carriers. They were hybrids for that. And so their child ended up getting that disease. What are the odds that they would? Well, you could do a Punnett Square to figure that out. When you cross two hybrids, what are the chances you would get two recessive alleles, 1 in 4? So there was a 25% chance they would have a child that had that actual disease, right? Now, if this were a dominant trait, that wouldn't happen, right? Because you couldn't mask a dominant trait with these two parents. Then one of them would have to have it if their child had it. So why are you doing this? Well, because you can cross pea plants like Mendel did and Indian corn or whatever. You can't do that to people, right? You can't say, hey, I don't know what your chances are of you having a child with this disease, so have sex with this person and that person, let's see the offspring. It doesn't work like that, right? So what you would have to do is look back at the history of that family. What do they have? Let's trace it out, and then we can predict probably what it is and how that particular trait is inherited. So autosomal recessive disorders, what you will find, just like here, okay, this skipped a generation. Neither one of these parents had it, okay, but they were carriers for it. So somewhere back there, somebody had it, right? So autosomal recessive traits can skip generations. So this mom had this particular disease. Dad, we don't know it's his other genotype, but they had 2 girls. Okay, both those girls are heterozygotes. We know neither one of them have the disease, but we know they're each carriers for that disease. This person married another person. We don't know what their second genotype is, but none of their children had it. But we know when we look at their next child down here that this individual, this son, must be a carrier for it because they produced a child that did have that disease. Now, if these 2 children marry or whatever, make children, then we could maybe figure out what their second allele is, but we really don't know. Now, this is another reason why you don't want to do inbreeding. This is why you don't want to marry a sibling or a blood cousin, because the chances you're carrying a whole bunch of recessive diseases within your recessive alleles that maybe are negative traits. And when you marry somebody in your family and have children with them, the likelihood of you producing a child with a combination of that is going to obviously increase. Okay. So when you look at autosomal patterns of inheritance, which is what we are on right now. We're not talking about sex chromosomes. Let's put a couple things down in our notes for 11.3. Autosomal patterns of inheritance. Autosomes are any chromosome other than the sex chromosome. Any chromosome other than the sex chromosome. Both males and females are affected with equal frequency. So you're not seeing boys or girls having more of one than another. And in a pedigree, females are circles, males are squares, and affected individuals are solid colors. For autosomal recessive, like the typical autosomal recessive patterns, most affected children could have unaffected parents, right? So when we saw this back here, okay, you could have people who have the disease, but probably neither one of their parents have it because it's a recessive trait, and you have to inherit 2 bad alleles in order to get that. Heterozygotes have an unaffected phenotype. So this individual, you know they're a heterozygote because they don't have the disease, but you know their mom gave them that A, that little letter A. So they are unaffected by it. And 2 affected parents will always make affected children. So if you have 2 people, if it's little a, little a, little a across, then the only thing they're going to be able to make is 2 little a's. All right, so some examples of autosomal recessive diseases are Tay-Sachs. This is a terrible, terrible disease that affects children. You usually only live till about the age of 4. And it's because they're unable to produce an enzyme and their central nervous system degenerates. So it's a progressive mental deterioration. The babies are born normal, but over time they degenerate. Blindness, paralysis, they can't hold themselves up. That is one example. So if you look underneath examples of diseases in the notes, I put Tay-Sachs down there as one. This is Tay-Sachs lacking the enzyme. Another one is cystic fibrosis. We talked about that earlier when you have channels that are not the right shape, and so the chloride ions can't move. That's another example of a recessive disorder. In cystic fibrosis, you remember that mucus accumulates in different, not just your lungs, but other organs as well. And these gentlemen are wearing percussion vests to kind of break up the mucus so they can spit that out. And then they're breathing in higher levels of oxygen because they can't pick up the oxygen as well. Okay, so that is another one. PKU is another one, and this is one where you don't have the ability to digest phenylalanine, and so that can cause problems. So the way they resolve that is they don't allow babies, if they get tested, they test them to see if they have PKU. They won't feed them food that has phenylalanine. So by altering their environment, you alter the expression of the gene, and we'll be talking about that a little bit later as well. Okay, so PKU, and then another one is sickle cell disease, and this is where your red blood cells sickle, because you have abnormally shaped hemoglobin, and that's just due to a point mutation, one wrong amino acid. So now we're going to transition into some dominant disorders, and one of those is Huntington's disease, and sometimes you can have signs as a teen, but a lot of times Huntington's disease, it's a dominant disorder, but it doesn't start expressing itself until you're like in your 40s. So that's a bit of a bummer, right? And so what can happen is you can already make children and pass that on to your children and not even be aware that you had it, right? But to have, if you look at this right here, this would fall in line with a dominant disorder because both mom and dad have it, but they didn't give it to their child. So what does that tell you about mom and dad? Mom and dad must be hybrids for it. So they're carriers for that, a good gene. They're carriers for a good copy of the gene, and there was only a 25% chance that they would give them that good copy of the gene. There was a 75% chance that this child would get Huntington's disease. 25% that they would be homozygous dominant, 50% they would be heterozygote. So hopefully you remember that from earlier discussions. So that would be an example. So when you look at pedigrees for dominant diseases, this is one that is called hypercholesterolemia, and this is high percentages of cholesterol, and there's associated problems with that. But what you can see in dominant disorders is that if you don't have it, right? If you don't have it, you're not like, let's look here. Okay, so dad has it. Mom doesn't have it. And we know a little bit about dad's genotype, and this would be something you could be asked to predict because they had children. So they had this boy here. So they had, in generation 2, they had 1, they had individual 3, they had individual 6, and they had individual 8. Okay. And because they had individual 3 and they had individual 6 who didn't have the disease, dad is hybrid for it, right? He is a carrier for a good copy of the gene. That's how they could make individual 3 right here. Okay, and 6. Now, if you look at individual 1, their first son, this son's genotype as well, because they have the disease. So they got let's say big H, but mom was a carrier for the little h because she didn't have the disease. So this dad is hybrid. He married a woman who doesn't have it. They have one child that has it and one child that doesn't have it. You could predict all of their genotypes just from working back and forth and looking at the inheritance pattern. So here, what you can see is autosomal dominant disorders don't skip generations. Okay, if this girl, right, these two parents both had this disease, right, and they had 4 children, 2 have it, 2 don't have it. This daughter married a man who doesn't have it. Okay, they don't have to worry about any of their children getting it. When it's an autosomal dominant disease, if you're clear, you're clear. You can't hide that dominant trait. So on autosomal dominant disorders, you want to have affected children will usually have an affected parent. I'm going to give you some exceptions to Mendelian inheritance in video 3 on this series, and that's called penetrance, and that is how often is a gene expressed. So there could be exceptions to that. So affected children will usually have an affected parent. Heterozygotes are affected, like this individual here. Heterozygotes are affected. 2 affected parents can produce an unaffected child, right? So in this case that I was looking, let's go back here, right? 2 affected parents could produce a child that's unaffected. There's only a 25% chance, but it could happen if they're both carriers for a good copy of that allele. And 2 unaffected parents will not have an affected child ever. And then I'm giving you some examples of those. Okay, neurofibromatosis is an autosomal dominant disorder. You get a tan spot right here. You want to watch those, especially those that can be exposed to the sun, because they can develop into tumors. The next slide's a bit brutal, so close your eyes if you don't want to see it. This woman had severe neurofibromatosis, and they started turning into tumors all over her body. Okay, I'm moving it now. Okay, Huntington's disease, like I said before, Huntington's disease is loss of control of your body. You can't hold your body still. You literally have to be tied down so you don't fall out of a chair. That is an autosomal dominant disorder as well. And then achondroplasia is the most common form of dwarfism, and that is an autosomal dominant disorder. So if these 2 married and they each had a normal height parent, and they have a good copy of the height allele, or a normal copy of that, they could have a normal sized child. And that has happened where 2 dwarf parents have a normal sized child because they were each a carrier for a good copy of that allele. I'm not saying dwarves are bad. Please don't say that. Okay. And then the next pedigree is an example. Is it an example of a dominant trait or a recessive trait? I'm trying to test you out here a little bit. And then I want to know, what is the genotype of individual 8? Okay. So do you think this would be typical of an autosomal dominant or an autosomal recessive? Okay, you thinking? Okay, so I would say it's an autosomal dominant. Why? This trait is all over the place. It's not skipping any generations. It's getting continually passed on through the offspring. And so that would lead me to believe that it is an autosomal dominant trait. That's what I would write for my null hypothesis. And then I asked you about individual 8. What is their genotype? Well, I'm going to look at this girl's parents. This parent doesn't have it, so they would have to have two recessive alleles. So let's just use letter A or letter A's. So that means that this individual is little a. So they would always give a little a to individual A. And they have it, so we know they would be big A, little a. They would be a hybrid. Now, we know what her father's genotype is for the same reason as what we just talked about before. So you could identify the different individuals and the different genotypes. Okay, so next video will be beyond dominant recessive alleles, and we're going to look at all the multiple exceptions to Mendel's law of dominance, segregation, and independent assortment. And if you're one of my students, I'll see you in class.